Hello and welcome everyone to the May 2023 lecture in our Paleo-Indian series. I just want to remind everyone that we will be recording this evening. We'll make the recording available for everyone afterwards. You can put questions in the chat box or the Q&A at any time. We will get to them at the end, as many as we can. And now I'll turn it over to Terry Wilson to introduce our guest and to thank everybody.
Good evening. On behalf of the Avon Historical Society, Avon Library, and the Avon Senior Center and the Planning Committee for Unearthing History, I welcome you to the third webinar in year 3 of the educational and informative events, mostly covering the paleo people, animals, and land of 12,500 years ago in this region. For those of you joining us for the first time tonight, we welcome you. For those who have been with us along this journey, welcome back. We thank our partners, the Avon Land Trust, the Farmington River Watershed Association, and the Institute for American Indian Studies in Washington, Connecticut. This year's series is sponsored again by a grant from the Lower Farmington River and Salmon Brook Wild and Scenic Committee. The Farmington River played an enormous role in the life of Indigenous people in this area, so we are grateful for the grant support. The origin of this webinar series is the discovery in 2019 of the Brian D. Jones Paleo-Indian site along the Farmington River here in Avon, Connecticut. To learn more, please visit the Avon Historical Society's website and drop down the page for the Brian D. Jones site. About 18 months ago, it was revealed to us in an earlier webinar that blood residue on a stone tool found at the Brian Jones site was discovered to have DNA on it from the elephant family. That told the archaeologists that it is certain that mastodons were present along with Indigenous people who were here. That discovery led us to our speaker tonight. So I want to warn you, she has a very long title. Dr. Heather M. Rockwell is Assistant Professor of Anthropology in the Noreen Stonor Drexel Cultural and Historic Preservation Center, which is in the Cultural, Environmental, and Global Studies Department at Salve Regina University in Newport, Rhode Island. Now, why is all that important? If you are not aware, about 75 years ago, the Sisters of Mercy were gifted with a mansion house as a base to founding the college, which encompassed 76 acres of historic grounds and structures. What excites historians like us is that Salve Regina University today has a very strong mission of historic preservation while allowing new technology to lead a diverse student population in research of cultural and historic significance. Hence, Dr. Rockwell's research in the microscopic analysis of blood residue on ancient stone tools is perfect for our webinar series. Dr. Rockwell has a bachelor's, master's, and PhD in anthropology from Beloit College in Wisconsin, University of Tulsa, and the University of Wyoming, respectively. Please welcome Dr. Heather Rockwell.
All right, thank you so much for having me. I am going to share my screen so you can all see this, and we will get going. All right, thank you so much for being here and inviting me. It's so nice to see such an engaged group. So what we're going to be talking about today is actually 2 different forms of analyses. My talk's entitled The Big Importance of the Small Things, Microscopic and Blood Residue Analysis of Ancient Stone Tools. So what we're going to cover today is basically we're going to define what are microwear and blood residue analysis, how are they conducted, some of the limits, and also some of the controversy around using these techniques, because archaeologists like to argue about everything. And the methods we use are included in that. And then I'm going to talk a little bit about some applications of these, mostly focusing on sites in New England. So I'm going to talk about the Potter site in New Hampshire, as well as the Jefferson site complex. And then just for fun at the end, I'm going to talk a little bit about the Laprelle Mammoth site that I worked on in Converse County, Wyoming. So without further ado, we'll get into our topic. So a really fundamental question in archaeology is, what are stone tools used for? And this seems really simple, especially if we're thinking about something like projectile points, right? These are kind of the creme de la creme of artifacts. These are what we get really excited about. And when we look at projectile points, we say, I know what that is. It's either an arrow or it's a spear, and it was used to kill animals. It's inherent in what we see. We feel like we understand it. But I actually argue that basing what you think a tool is used for by what it looks like or how it fits in your hand is a really bad idea. Because people are creative creatures, right? We're the clever monkeys. And we pretty regularly use tools for something way beyond their intended purpose. So take, for instance, your standard flathead screwdriver. Based on the design and its form, it has two jobs. It's either to place or it's to remove flat-headed screws, right? That's all we use it for. But we use this tool for all sorts of things it wasn't intended for. It could be installing an eye bolt or opening paint cans, stealing an older model car, or accessing parts of your computer that Dell really wishes you wouldn't access. So just because that's what the tool looks like it's used for, or that's what it was designed for, doesn't mean it's actually what it's used for. And this is even more important if the form isn't giving us any clues. So if I showed you this pile of lithic debitage, or the product of making stone tools, and I told you that in that pile of sort of unassuming rocks is a huge variety of tools, we would have a really hard time determining their uses just based on what they look like. Frankly, we would have a really hard time sorting out which of these actually were used as tools, and the ones that are just waste flakes, that are just the product of making other tools. And this is where analyses like microwear and residue analysis can be really powerful tools for archaeologists, because they can let us see aspects of human behavior in the past that might not be visible through other means. So later on in the talk, I'm actually going to talk about why in New England especially, that can be really important and powerful. So I'm going to start with microwear or use wear analysis. So microwear and blood residue analysis are actually two separate areas of analysis. So we're going to start with this one and then we'll move to blood residue. So micro or use wear analysis is this technique that we can use to answer the question of how were stone tools used. I was first introduced to this when I was doing my master's thesis work at the University of Tulsa, excuse me, by my advisor, who's actually one of the founders of this technique. But what we do is we basically examine the surface of a stone tool under a microscope, and based on patterns of breakage on the surface, we can see how that tool was used. Now, this technique, actually, interestingly enough, was first developed in Russia. It was developed by a gentleman by the name of Sergei Semyonov. He's a Russian archaeologist. He's living in Moscow in the 50s, and his archaeological lab shared space with a metal shop. This is kind of the classic thing with archaeologists. We always have our offices in really weird places. University of Texas had their archaeological labs underneath the football stadium. My office is in a converted stable block, and the University of Wyoming used to share space with a cheese lab. So it's not out of this world that his office would have been stuck in the middle of a Russian metal shop. But Semyonov basically got inspired. He's sitting in his lab and he's looking around at all these metal tools and these drill bits and these scrapers and planing tools. And he notices that as these tools are used over a period of time, they start to develop these tiny lines and striations on the surface. And these striations vary by how the tool is used and based on the material it was used on. You get different kinds of lines. And so, for instance, if we were looking at the striations that form on a drill bit, right? They're going to form in a spiral pattern, mimicking the way that's used. And he wondered if this same principle could actually be applied to stone tools to determine their use. So he did this massive experimental study where he looked at to see if this would actually work. And luckily enough, it did. He ended up publishing this volume that's called Prehistoric Technology, Experimental Study of the Oldest Tools and Artifacts from Traces of Manufacture and Wear. Now, it's first published, it's a really important book, and he calls it Traceology, and it didn't receive really any attention. This is probably because it was being published in Russian in Russia in 1957. We're in the Cold War. And so it really doesn't get any attention until it's published in English in 1964. And this initially kind of has a rocky start. It's picked up by a few archaeologists throughout the 60s. We get these early papers where people are kind of trying this out. They're trying to find these striations on stone tools to interpret their sites. But they're not having a ton of luck, and people really don't kind of embrace it all that much right away. But it emphasizes these early papers, the importance of doing these experiments. You basically need to create collections of experimental tools like the one you see here on the right to compare to archaeological collections. You get this kind of base knowledge. And it's not really until the 1970s that we see people really pick this up and run with it. And there are three big papers, the ones that I list here, that we kind of credit with founding modern microware technique. And we also start to kind of get the way we do it, and we start to separate and specialize a little bit into two different forms of analysis called low and high power. So this is Sergei Semyonov with Ruth Tringham, who's kind of really the mother of microware analysis in the United States. So I said the 1970s is sort of this birth of modern microware. And a big part of that is they expand what we're looking for. So Semyonov was really into these striations. But what later analysts discover is striations are really hard to see, and not all materials preserve them particularly well. But what they do find is that lake scars on the edges of tools and the development of polish, basically shiny surfaces on the stone tools themselves, can also be indicative of wear. So different kinds of polishes will form based on how the tool was used. And they also standardize the process through two big things. One is doing those experiments, and also doing a process that's called blind testing. Now, experimentation, this is the fun part, I think, in some ways, because it's really hands-on. So experimentation is sort of a required part of any sort of this analysis. What we've discovered is that the way that stone tools break is really strict, right? It governs these rules we call fracture mechanics. So stone tools will break in a particular way. But what we found is the way it develops those fracture patterns on the edge falls in certain categories based on how the tool was used and how hard of a material it was used on. So what's really important for microware analysts is we create these massive experimental collections. We basically use hundreds upon hundreds of stone tools personally, so we know exactly for how long it was used, on what, and sort of how it responded, to create these massive comparative collections. And a big part of this is we try to get as close to how people would have used them in the past as we can within the limits of kind of our modern lives. So for instance, when I was doing this at the University of Tulsa, I wanted to have a good number of butchery tools, but I was living in, urban Tulsa, which means, I can't go out and sort of butcher a deer on my patio in urban Tulsa. So a lot of times this involves us kind of getting sides of meat and doing small butchery projects in our house. So we get as close to real as we can. When possible, I always try to get people to do these experiments for me, people who really know how to use them. So for instance, I was working with some Peruvian colleagues, And they were working with basically an indigenous population living in the high Andes, and they wanted to butcher an alpaca. So they had the indigenous people use stone tools to butcher the alpaca, because that's going to get us as close to real, as close to natural as possible. That's the ideal, but we don't always get there. The second part is sort of a testing thing. It's called blind testing. And what this means is we have these massive experimental collections, right? And what's done is basically is to assess whether an analyst is actually qualified to do what they say they can do. So what we do is you have an outside party select a bunch of experimental tools for you to look at. So they know what they're used for, you don't. And then you have to identify where everything was used and what it was used for as specifically as possible. And then somebody comes in and they grade you. They actually look at, did you get perfect identification of where the use was located, the action it was used for, and the contact material it was used on. So it's, most people don't get perfect scores because people aren't very perfect. And they specifically usually try to make these very, very hard. So things like having materials or tools that are used on multiple materials, or slipping in a few artifacts that aren't utilized at all. The point is sort of to make it really, really hard and really, really tricky to show that you can really do this technique and you can do it effectively. So those are kind of the things our friends in the 1970s really established as, this is what all microware analysts should be doing as part of their analysis. But the other thing that we see in the 70s is the field splits into two directions. So one is called low power microware and the other is called high power microware. Now, low power microware is, as the name implies, looking at lower magnification, between about 20 and 150 times magnification. And it's really focused on those principles like fracture mechanics. So how it, as you can see in this image here, you see these little microfractures along the edge of the tool on the left. It's looking at those as the primary indicators of tool use. Now, because you're not looking as closely at the tool, you tend to get a little bit less detail. So I might be able to say that this tool in the picture here was utilized on wood. It was used to whittle a piece of wood. But I'm not going to be able to tell you much more than that. I can just say it's used to whittle wood. But because we're getting a little less detail, it means we can actually look at very large samples of tools and we can look at it pretty quickly. It also tends to be lower cost because we need a less technical microscope because we're only looking between 20 and 150 times magnification. High power, on the other hand, is again looking at really high power microscopes. So 100 to 500 times magnification. We're often using things like scanning electron microscopes that are giving us really, really, really minute detail. And they tend to be more focused on polish development. So you can see here this shiny surface on the tool and those little pits within it. They're looking at that as the primary indicator of tool use. Now, high power has some raw material limitations. So for instance, here in the Northeast, we see a lot of indigenous people using things like quartz, right? High power analysts can't look at quartz. It doesn't basically develop polish in a way that other stone tool types do. They, however, can offer, on the tools they can look at, really incredible detail. So for instance, where I said low power might only be able to tell you they worked wood with this. High power analysts could say they worked softwood or hardwood with this. So much, much, much more detail. But that means with that detail comes, it's way more time consuming. So they tend to look at pretty small samples of artifacts and they need more technical equipment to do it. So it's also, it's a little more high cost, but it's that, you know, you get what you pay for. So it might be nicer. Personally, I primarily use the low power microware technique because I often am looking at multiple sites to get sort of a regional perspective on what people are doing here. Now, this split in the field leads to what I call the ugliest debate in archaeology that no one has ever heard of. I said that archaeologists love to argue with each other. And this is especially true when we are talking about microware analysis. Basically, from the late 1970s through the 90s, low power and high power analysts spill a lot of ink screaming at each other that their method is the better method for doing microware analysis. And these articles get nasty. They really go after each other. One of my favorites was written by my master's thesis advisor, a gentleman by the name George Odell, who basically said, I don't know why we're all yelling at each other. This is crazy. And so he wrote this parable that's called Brer Rabbit Seeks True Knowledge. If you can get your hands on it, it's a very funny read. And it's basically criticizing how much microware analysts are sort of screaming at each other instead of really getting anything done. And for a while, this leads to actually low power analysis kind of falling out of favor. So people sort of are only using high power. Now, generally, these really nasty fights don't do great things for archaeology, but actually one of the small benefits from this is it sort of forces this level of academic rigor where people really have to be super good at what they do in order to be taken seriously. So it really makes microanalysts pretty strong. Now these critiques continue, and we see them kind of into the modern day. Because I think basically microanalysts yelled at each other so much that people outside of microware kind of got in on the fight. So from really its earliest days, microware has been met with really heavy suspicion owing to a few different things. So in the early days of the discipline, critiques are focused on things like small sample sizes. So if you're doing high power analysis, you might only look at 4 or 5 tools, which means people were like, well, these results don't really mean anything. They're kind of anecdotal. You know, it tells us what 5 tools on a big site are used for. That doesn't give us very much. It also there was sort of inconsistent reporting standards. And despite people using blind testing and giving these really detailed reports, a lot of people basically said, I don't buy this. And it took a really long time. And we're still sort of struggling for acceptance today in North American archaeology. So microanalysis is really big. The largest schools for it are actually in France. But in North America, there's very few analysts actually working within this field, I think partially because of so much kind of fighting. And it's also been viewed as sort of having a lack of innovation, that for a long time, we were kind of doing the same thing that they were doing in the 70s, and we needed to, get some new technique. Now, I think recently we've had much more advances. So one of the big ones is sort of the scanning electron microscope, which gives us these really kind of beautiful 3D images you see on the left. They're much easier to interpret, and it removes some of the kind of ambiguity we were dealing with in early microware. We're also seeing the application of laser scanning confocal microscopes. So basically, we're getting better tools to do the job we have, and it's helping to kind of gain more acceptance within the field. Now, more recently, we've actually seen some interesting applications in the field of photogrammetry. So if you're not familiar with photogrammetry, it's a pretty interesting technique. Basically, with just a camera and some fairly inexpensive software, we can create fairly detailed 3D models of anything, really. Statues, stone tools, artifacts. And people have more recently started applying this to microware analysis with an attempt to sort of quantify what we're seeing of, can we actually have a computer help us identify tool uses once we build these 3D models? What's exciting about this in particular is the applications are sort of endless, and it's really powerful and it's really cheap. Most of us have a digital camera nowadays. So if you hook it up to your basic microscope, you can actually make some really detailed 3D models of stone tools. All right, Now that I've given you probably way more detail than you wanted on the microwear side, we're going to talk at least one case study. So this first that I want to talk about that I did microwear on, this was actually part of my master's thesis and dissertation research, and it's the Potter site. This is located in way northern New Hampshire. So if you've ever been up to Randolph, New Hampshire, I'm sure most people haven't. So if your New Hampshire geography is lacking, that is north of Mount Washington. It's sitting right on Route 2. Now, Route 2 in the state of New Hampshire, archaeologists there call it Paleo Route 2 because there are a ton of paleo-indian sites found all along that road. Now, a big part of this is you guys are all from New England, so you know that east-west travel in New England is an unnatural act. We do not do it. We are north-south travel people. And that's because of the way the mountains run, right? They run north-south. So there's only so many paths through the mountains that keep you low. And Route 2 is one of these. So we think this became a pretty common travel corridor for paleo-indigenous people. Now, the Potter site is an early to middle paleo-indian site. It dates between 12,750 and 11,600 BP. Important thing to know, though, is there are no organics from that site. There is no carbon. We have no radiocarbon dates. So, and the dating we do have for that are basically coming from the two projectile points you see here on the screen. The one on the left is a Bull Brook point that is an early paleo-indian point. And the one on the right is a Michaud-Neponset point, and that is a middle paleo-indian. So that's how we got those dates. But literally everything we know about this site comes from stone tools and stone tools alone. We have no other materials recovered here. Now, the environment of this site, I think is really cool. During the period of occupation, this site would have looked really different from this photo you see here. This is actually looking over where the site would be located. So for the post sort of ice age landscape, for one thing, we have to remove most of the trees. So northern New England in particular, at the end of the ice age, doesn't have very many trees. It's what we will call a spruce parkland environment. Now, there aren't really good analogs for what the environment would be like, but think more like Newfoundland. All right, kind of open. There are trees, but they're really kind of focused around water sources like small kettle ponds and rivers, a much more open space. It would have been a much drier and much colder environment. And because of this, it would have been able to support animals, in particular, caribou. We think caribou were a very important resource for paleo-indigenous people throughout this region. And obviously, as you all know, for Brian G. Doan's site, megafaunal species like mastodons were also probably being hunted. We don't have too many examples of sort of mastodon finds in general. And obviously, Brian D. Jones is really the first that has a mastodon blood residue associated with tools, but we know they're here. So this would have been a really different environment to people to live and make their life in. So this is sort of giving you a little bit of a bird's eye view on where the Potter site is. You can see that it's down in the valley. It's really sitting in the shadow of what's called Mount Madison. And that road you see in the picture here is Route 2. The small blue dots you can see right by where the kind of line comes down, those are a series of small kettle ponds. Now, the excavations we did at this site is absolutely massive. We started excavating here in 2003 when I was an archaeology baby. I started working at this site when I was, I believe, 17 years old. It was excavated as part of the New Hampshire Archaeological Society and the New Hampshire State Conservation Rescue Archaeology Program, which is a really amazing public archaeology program that was willing to take me out with them. We did a massive number of shovel test pits on this site from 2003 up until about 2012. So the area of the site, when you're looking at the map here on the right, the gray area is a hectare in size. It means 100 by 100 meters in area. And every dot that you see in that picture represents a 50 by 50 centimeter shovel test pit. So we hit this site really hard. The red dots on the screen are those that produced artifacts. So you can see lots and lots of artifacts, but what's important to note is also the black dots. Those are all the areas that didn't have artifacts. So it's not like it's one consistent smear across the whole area where there's just artifacts everywhere. There's empty space. And that can be really important for understanding the past, because it gives us the opportunity to maybe look for things like structures or different periods of encampment. Because as I said, we have those two different projectile point styles, which means we know people were using this site repeatedly over a series of years. So the areas you see circled on this map we refer to them as blocks. These are areas where we found really dense concentrations of tools. And so we opened up sort of larger excavation areas. Now, in almost a decade of archaeological work at this site, some 11,214 artifacts were recovered. It's a massive number. Now, because this site contained only stone tools, in order to better understand how it was used, I conducted a micro-rare study on this. I examined all of sort of those formal tools like our scrapers and projectile points, but I also did a random selection of pieces of debitage, just in case to see if these were used as tools. Now, I examined a total of 1,000 artifacts. And of these, I was actually only able to identify 94 total pieces that were utilized. Now, you might be like, well, she's not very good at her job. It wasn't that. It was because this area, we said there's no preserved organics, right? The soil at this site is so acidic that it actually was causing what's called desilicification, meaning the silica, the part that makes stone glassy, was actually leaching out of the stones, and it was turning them basically into powder. There were some artifacts that by the time we got them back, they were all smoothed over their whole surface because basically just the friction from the artifact bag they were in wore down the surface of the tools. They were that friable. And this didn't seem to be evenly distributed across the site. So some tools were in perfect condition, could be analyzed, and then some were kind of a mess. So we were able to get only a small sample. But what I found was really, really interesting. For one thing, the utilized pieces were split evenly between, so I found 47 of the formal tools that I looked at showed evidence of being used. And I also found 47 pieces of debitage that were utilized. Now, one of the things that's kind of interesting is that the expedient tools are really clearly an important part of the toolkit. And these are things that we might miss if we weren't conducting this kind of analysis, because it's really important to see that the expedient tools are getting used for different tasks that are more formalized items. So we see here our expedient tools are being used mostly for butchery tasks, what I call here longitudinal soft activities, whereas formal tools at this site seem to have been primarily used for tool creation and maintenance. In particular, they're doing a lot of woodworking at this site. So our hypothesis is that this site was located on these kettle ponds, may have made it a good location to resupply your wooden tools. So we said that the area is more open, there are kind of few tools. This may have been a spot where there were good wood resources, as well as access to kind of animals that people would want to camp here. And this also gave us some indication of when this site was used. So for one thing, we know that they reoccupied the site. They came back to the site again and again and again over a period of several hundred years. But regardless of when they were there at all of those different localities I showed on the map, they seem to be doing lots of woodworking and lots of what I call kind of tool maintenance tasks. this is sort of things like bone working, woodworking, sometimes like small soft cutting, probably things like sinew. And I think this is related to what we call gearing up. Basically, you are focused on getting your toolkits in a place where you can go out and do something really important. So we said that paleo-indigenous people, particularly in the North Country, are hunting a lot of caribou. So what I think is this is probably a late summer occupation where people are getting ready to go hunt caribou when caribou come through in their fall migrations. We know that caribou during certain times of the year group up into these massive, massive, massive herds, and they would move through the area as a big group. So what people would do is they'd position themselves on the landscape where they could hunt those caribou and kill lots of them all at once to get what they needed for the rest of the year. Things like meat, but also as importantly, things like hides. It's estimated that one person needs 35 caribou hides per year in order to have all of the clothing and equipment they need. So if you're going to need that many caribou to support your population, you need to be really, really prepared when they group up in these big, massive migrations. And that's what we think they were doing at Potter, is that they are getting ready to go attack these caribou. Oh, sorry, just having a little problem. Tried to go too far. There we go. All right. So that's microwear. The second technique is blood or protein residue analysis. And many people think that residue analysis and microwear are the same thing, but they are actually different. So microwear uses, again, just looking under a microscope, straight up, using your eyes to see what's there. Blood, or what's actually called protein residue analysis, is used to identify what our stone tools are used for, but using chemistry. So it's actually called protein residue analysis in most sentences, but colloquially we call it blood residue analysis because that is most often what we're looking for. So it's a pretty different process from microwear. So microwear is my primary research tool as an archaeologist, and I can do that myself with a microscope. But residue analysis is primarily doing lab-based chemistry. So while I often identify the presence of a residue on the surface of a stone tool, like the one you see on the right here. So you can see here on the far right, this kind of darkened section on that very light tool, that's a residue. But that's kind of where I stop. I don't know what that residue is. So in order to get more information, I need to send it out to do a lab analysis. Now, to give you a little bit of background on kind of where this technique comes from. So unlike microwear that actually, was created by archaeologists, residue analysis was created by physicists and chemists. In particular, Abelson, Hare, and Horing at the Carnegie Institute started to develop this technique again in the 1950s and 70s. We're actually doing like a ton of really interesting scientific research during that time period that I feel like doesn't get enough credit. And their work this time is sort of paralleling the early work that's being done to understand DNA. But what they and others basically learn is that amino acids in protein can survive on the surface of things for a really long time. So much of this early work is actually conducted on shells because archaeological sites that have shell in them often have tons of shell in them. They'll have massive, massive quantities of it. But what we see is that a lot of times shells are really fragmentary, right? Shells break up pretty easy. So it's sometimes hard to identify if they're, marine shells or shells that are coming from sort of river sources. So what they did is they were sort of testing this residue analysis and this amino acid study to see if, to sort of reconstruct the paleontological record. So now this takes a big jump forward between the 80s and the 2000s. So we can, again, residue studies are sort of paralleling the development of DNA studies. In that as more and more scientists work on this problem, we're able to use sort of smaller and smaller samples and get more refined results. So DNA and protein could both be used in theory to identify a species, but there's a key difference here in that DNA is obviously analyzing sort of our core founding blocks. Whereas when we're doing protein analysis or blood residue analysis, what is actually preserved is we're looking at antigens. So I don't want to get too deep in the weeds in chemistry. I don't want anyone having flashbacks to high school. But this is also sort of used in things like zooarchaeology through mass spectrometry. So how we're doing this is you do low power microwave first, and you identify the presence of a residue. So for instance, that's something I would do. I would sort of describe what I'm seeing in the photo. Then that is taken and they actually use a scanning electron microscope to identify the presence of red blood cells. So we see a residue on the surface of that artifact. And to identify red blood cells, the red circular thing you're seeing in this upper image is an antigen. And so you have these in your body. So we use this ultrasonic chemical bath once we've identified that there's blood there, and it removes them from the surface. You basically get a liquid. Now, the Y-shaped thing you see on the surface here is an antibody. And antibodies will only cling to antigens that match with them. So this process happens in our bodies when we have an invading disease antigen. Our body produces antibodies that are specific to that disease for our immune system to function. So for instance, if you had chickenpox when you were a kid, right? Your body, so chickenpox was the antigen, your body produced antibodies that were specific to fighting chickenpox. But those antibodies, those little white Y-shaped things here, will only bond onto chickenpox. So if you introduce smallpox into your system, it doesn't know what that is. Those antibodies won't fight that different disease. We use this exact same principle to identify different blood types. And I said this can be better in some ways than DNA, or at least a little bit easier for our purposes, because proteins, these antigens, can remain on the surface of an archaeological or a paleo-ontological material for a very long time, up to 1 million years and possibly longer. This is pretty controversial, but they're starting to do some really interesting work with dinosaurs, actually. So way before anything we're concerned about, but 1 million years is considered pretty stable. And they can preserve on lithics like scrapers and projectile points. But these have also been recovered on groundstone tools and even soil samples. So we can get this really broad picture of the past using even things like the dirt. Now, the biggest drawback of this is it can often only identify species to the family level. So for example, if your sample returns camel proteins, right, camel blood, it could be any camelid species. So that could be llamas, alpacas, vicuñas, or it could be camels. But, you know, that's basically as detailed as we can get. Now, a lot of times this maybe isn't a problem, right? If you say camelids, you're like, yeah, I feel pretty comfortable with that. I can guess based on what species were available, which one it is. But sometimes when we're making really big claims, so for instance, if I'm working here in North America and I have camelid protein and I want to argue that they were hunting camels in North America, camels go extinct really early, right? That would be a big argument. Some people might be like, well, how do you know it's not a llama or an alpaca? So it can make it a little hard to take a big swing and make a big claim. The other thing you might be saying is like, well, why not just test everything, right? Throw a ton of these in. That way you can really, if you just test all the stone tools you have, so you can see all the different kinds of proteins, that would be awesome. But it's pretty expensive to do. It's about $200 a sample, and you get a lot of negative results, right? It makes sense. It's not like there's blood on everything. There's not proteins preserved on everything. So we can't really test everything under the sun. When you submit these, it's a little bit of a gamble. Now, for an application of this from my own work, we're going to look at the Jefferson Site Complex. Now, this is just down the road on Route 2, headed west from the Potter site. So it's in the Jefferson Village. If you've ever gone to Santa's Village, you drove right by where this series of sites are located. This is a complex made up of 7 different archaeological sites. They all date to the paleo-indigenous period, and many of them appear to be sort of small campsites. Now, the one we're concerned with is called Jefferson 6. This site, we were able to conduct a really massive excavation. This was essentially a recovery project. The owners of the B&B that you can see in the background here needed to put in a new septic system, and it was going to go right in the middle of our archaeological site. So luckily, Dr. Dick Bovair with the state of New Hampshire was able to convince them to give him a little time and let us come in and do a huge excavation at this project to recover as much as we could before this site was destroyed. Now, this site produced, again, tons and tons of artifacts, many thousands, but much like the Potter site, this contained no carbon or preserved fauna materials, just stone tools. Now, I've done a limited amount of microwear on this, lots of scraping activities, but it seems to be, I saw a lot of hunting activities at this site. There seem to be lots of tools being used for butchery. And so because of that, they decided to send in a tool that I had identified as a butchery tool for blood residue analysis. So it's this tool here on the right. It's sort of an unassuming kind of blade-like flake. So it was sent out to the lab. They conducted blood residue studies. And what returned was the presence of bear protein. Now, this is sort of surprising for us because paleo-Indian research in the North tends to be really focused on caribou hunting. And we can sometimes forget that caribou are a seasonally available resource, mostly in the spring and the fall. So I said they do those big group ups. Well, when they're not grouped up, they're actually really dispersed and they can be really hard to hunt. They're not a very reliable resource. So it's likely that paleo-Indian people would have needed to supplement with other game on the landscape. And bear is one we often don't think about. So this result was really important because it sort of expanded our horizons in terms of the kinds of animals that they would have been hunting at this time. We would have totally missed this without blood residue analysis. Now, just for fun, I had to slide one in. I worked in Wyoming is where I did my dissertation work. I lived there for about a decade after I finished my dissertation, working for the state of Wyoming and working on some research projects around the area. And one of the projects that I had the most fun on was the Laprelle Mammoth site in Wyoming. This is a deeply stratified mammoth kill site. Sorry, I had to take a drink of water. It contains massive numbers of stone tools. It has a huge red ochre stain across the surface of it. And most importantly, it contained the remains of a Columbian mammoth, as well as several small bones of different kind of faunal remains. Now, I was lucky enough to get to conduct the microwear study, but what was really interesting was the blood residue analysis was sent out to a colleague of mine, Dr. Robert Yohe, and we did not communicate during our analysis. So he did the blood residue, then the artifacts were sent to me, and we didn't talk to each other about what we found. We sent our results to sort of the principal investigator on this, Dr. Todd Suravel, and then he kind of put them together. So it's sort of a double-blind study. But what was exciting is that our results lined up really, really well. So I identified a large number of tools that were utilized for butchery and hide working. And Yohei's results identified several faunal species, primarily elephant, unsurprising, right? Because we were looking at a mammoth. But he also identified things like possible swine and potentially horse within the blood residues, which is pretty interesting. So by doing these double-blind studies, we were able to have much more robust results for the paper and confirm that there was this really big breadth of activities at the site. Now, this was really important for this one because previous work had argued that this was not even a human occupation. They had argued that these stone tools weren't associated with the mammoth. So basically, the blood residue was able to confirm that no, these stone tools are associated with the mammoth because they actually contain mammoth blood. So these technologies can really be amazing tools for enhancing our understanding of the activities at an archaeological site, in particular sites where there's very little preservation of organic materials. So for places like New England, this really matters. We have pretty unfavorable geochemistry around here, so we rarely get preserved organics. And so these techniques might be our only opportunity to see the soft portions of people's lives. We as paleoarchaeologists, we tend to be a little guilty of being sort of stones and bones archaeologists. But the majority of people's lives in the past are not made up of stones and bones. So for instance, if you look at this image of an indigenous family outside of their home and you look at what's in this picture, we don't really see any stones and bones. We don't actually see anything in this picture that would preserve at our archaeological sites in New England. So techniques like microwear and residue analysis might be the only ways we could actually see these portions of the past that really made up the majority of these people's lives. And that's really what we want to get at, sort of how did people live at this time period? So with that, I just want to say a quick thank you to the New Hampshire Archaeological Society and New Hampshire Scrap for giving me access to these collections, the University of Wyoming and the Friesen Institute for supporting my research on those, and to the Avon Historical Society for putting this event on. Having this larger group of engaged, interested people in archaeology is really special and spectacular. So I'm truly honored to have gotten to speak to all of you. So I will stop sharing.
Fantastic. Thank you.
Thank you.
And we do have a couple of questions that have already come in. All right, so the first one is, why hasn't anyone used archaeological geophysics on the Potter site to try and identify archaeological features that would provide better context and dating for the lithic assemblage?
So that's a great question. So GPR is sort of a big one that people use a lot for geophysics. It's really bad at finding fire hearths. We have been experimenting with some magnetometry, but to no avail. It really hasn't worked well in there. Part of that is the context of that site. So it's in juvenile forest. And so it's all little dinky trees. So when we worked at that site, we actually had to have somebody, my husband, on full-time chainsaw duty just to drive lines through the forest. And because of that, we've got a lot of root action, a lot of things kind of moving around. So we know there isn't very good, in particular, vertical integrity at that site. So geophysics hasn't worked really well. Now, my current research project is in northern Maine at the Munsunging quarries, and we have had some success using magnetometry on that site. So we may have some applications on paleo-indigenous sites. That's a really great question.
Okay. The next question, can you process any stone tool for protein analysis, or does the tool need to be handled in a particular way when it's found? And can handling it by modern humans contaminate the tool?
So if you were doing DNA analysis, we would be like, you have to be crazy, crazy, crazy careful. Protein analysis is a little more stable. You still have to be careful. So the big one is, I usually tell people if I'm going to do microwear, if I identify any sort of a residue, I'm super careful with it. I don't wash it. And a big one is many microanalysts will put things in what's called a sonicator. So it's basically like hyper cleaning it. If you did that, those residues are gone.
Yeah.
So if we see anything on the surface of a tool, I often will label them do not wash. And that's why you can be like, most of the washing I do, I use like a toothbrush and I sort of gently clean them. That residues will actually sometimes hold up through that, but it's harder to contaminate them, luckily. DNA, real easy to contaminate, but proteins are a little more rugged and a little harder to contaminate.
Okay, and then following up with the DNA part, have you ever, or do archaeologists ever find human blood on any of the proteins in analysis, or even DNA? So how do we know the humans were there is really the question.
Yes. Yes, they have done blood residue studies that have identified human proteins. So one of the sites I worked on in South America, so I did some micro rare on a South American site, that they argued that it was the earliest evidence of beheading, because they had stone tools that had evidence of butchery wear on them and also had human protein on it. So yes, they do.
Okay. Okay. If anyone else has any questions, please type them in. We'll happily read them. Another question on the Potter site. Can the Potter site used for tool gearing up provide any indication for how close kill sites would be?
So I have opinions on this. I love that question.
Okay.
So yes and no. So that Route 2 corridor that I was talking about, that it's the travel corridor for people, animals and people like the same kind of travel corridors, especially caribou, right? They're not going up and over the mountains. So my thought is, if you're kind of camping seasonally in this location, you're sort of gearing up, you're going to do that close to where you hunt caribou. So I think it's probably not that far away. We should be finding some sort of a caribou kill site. Similar, if you're at all familiar with the Vail site in Maine. So the Vail site in Maine, I did the micro rare on that, and I argue that that site is without a doubt a fall camp, a fall caribou hunt, because they have tons and tons and tons of scrapers that are all basically used to work hides.
Yeah.
If you're doing that, that's a fall caribou hunt. We should be finding a site that is similar to that, fairly close to the Potter site. I would argue within probably 20 to 40 kilometers. So my dream someday is to do a big survey all along Route 2 and just like go find some cool sites.
That's it for the Q&A that has come in right now. Terry, well, I was gonna say, before we turn it to Terry for closing thoughts, is there anything you want our audience to know that either came up through the questions, or just a parting piece of homework, or anything you can give us on this topic, or that you just want us to remember as we close?
Big takeaway. So micro rare, blood residue, different things, but they kind of have parallel goals. It's really kind of, for us here in New England, it is one of the few ways we kind of get to see stuff that's past the stone tools, because we have kind of crummy preservation. So it can be a really powerful tool for analysis here. And I hope to see more of those studies. I think it's great that they've done that at Brian D. Jones.
And did you say earlier, when before we signed on, did you say you worked at the Templeton site here in Connecticut?
I did. Yeah, I did the micro rare for Templeton. So that publication is pending.
Okay, all of our viewers are from that site. I mean, they've worked on that site because they're from the Institute. So I see they're on. So I just wondered if you wanted to share anything, but if you can't yet, that's okay.
Templeton is really cool. It's such a neat site. Really diverse range of activities that sort of support the interpretations that we have of that site, that's a habitation site. I think it's a slightly longer term occupation where people are kind of hanging out. It has some indications of being kind of a gearing up site, but it's got a little bit of a different flavor to the Potter site. I think it's more, we're seeing a little bit more of kind of aggregation behaviors there. So I think this is people getting together and doing sort of collective labors, which is really cool.
Well, your final thought has generated a couple more questions, which I sort of love. So, is there an overlap in these type of archaeological protein analysis and criminology techniques?
Yes, there are. Developed for the, archaeologists, we borrow from the best. We have very few things that are actually ours that originated with us. All of these techniques are really being borrowed. Anything that's coming out of like chemistry and physics, it's usually made for like solving crimes first. And then we're like, there's an idea. And this is no exception. Sort of really early on, before they were doing DNA stuff, this is what they were doing to identify blood.
Okay.
Is identifying, they're like, oh, yep, this is those. If you watch like old like British crime procedurals, and they do like this quick little test thing, and they're like, oh, it's blood. That's this.
Okay. One of our guests is from the Mid-Atlantic, and she said their geology is similar to our area in terms of the preservation. Do you have any suggestions for finding? Hold on, I just lost. For finding a source who can do microware analysis in the Mid-Atlantic.
Tom Label is a very well-respected microware analyst in that region. He did work on Shawnee Mini Sink, if you're familiar with that paleo-indian site. He did all of their microware. He is spectacular, a very nice gentleman, probably one of the busiest people I know, but he is an excellent analyst. Ashley Smallwood does stuff in the kind of the southeastern portion of the United States, but going into the Mid-Atlantic. She is also a top tier microware analyst. She mostly does high power analysis, but she is great to work with, and can give you really great detail. Those are two of the colleagues I know who are working on sort of paleo-indigenous stuff in particular. Tom is a little more diverse. He does a lot of stuff, but Ashley is a paleo-indigenous specialist. And they both work down in that region and are great.
Okay, so thank you. And then back to the tools, there's a couple of questions with it. One is gender themed in terms of, is there any blood found on what might be considered women's tools?
Yes, definitely. So controversial theory. My dissertation advisor was a woman by the name Nicole Wagisback. She has this paper that she basically says that the more meat you have in your diet, what we see ethnographically, when we look at cultures across the world that were hunter-gatherers, the more meat you have in your diet, the more non-hunting tasks are done by women. So basically anything that's not directly hunting related, like going out and actually killing the animal, is done by women, because men are spending pretty much all their time going out and hunting, the more meat you have in the diet. This includes things like butchery, and this includes all of hide processing. Hide processing activities, ethnographically the world over, are women's work, which means things like scraping tools that are used on hides, those were probably operated by women. They were also probably made by women, because if you think about it, it doesn't make any sense that it's like, well, the husband's out hunting, I guess I have to wait for him to come home to make my stone tools for me, right? That doesn't make any sense. You're going to be able to make your own. She actually argues that this might also mean that women were involved in making fluted points.
Ooh, okay.
Because the more men hunt, the more women are actually the primary tool makers. So kind of a fun idea.
That's a great theory.
Okay. Is there any difference in the quantity of the blood residue between the kill points versus the butchering tools?
Not necessarily. Honestly, we don't have particularly representative samples, so it's kind of hard to tell. Like I said, they're $200 a pop, so you might only send in a few objects. So you don't necessarily have a particularly even distribution. So it's hard to say, but from what I've seen so far, no, not particularly.
What, if any, would the role of AI be in making this analysis either faster, less expensive, more accessible, etc.?
In terms of microware, I actually think there's some really interesting potential. We've been looking at sort of ways to so microware kind of has this issue where it's sort of seen as wizardry, because you're interpreting what you see, right? Like you see this pattern and you compare it to another pattern, and humans are really good at that, right? Like pattern recognition is one of our superpowers. We're really good at matching games, but it feels a little subjective. And a lot of archaeologists, they like their archaeology with a side of statistics. So we've really been looking for ways to make microware more quantifiable. And AI is one of the areas that people are like, this is interesting. This could potentially give us that path where you sort of feed in sort of the visuals that you have and let it do that pattern matching for you.
Okay.
We just have to find more computer programmers, because boy, is that not me.
Okay. All right. That actually got us through. That is the end of the Q&A. So now, Terry, if you want to close everything out, you can.
Yes, I want to thank everybody again for participating with us. And we do have 2 more coming up this fall. We may throw a couple more in like some other hot topics that are coming up towards us. And please, if you do have any artifacts that you have found that you think might be native to this area, please sign up to bring them to Artifact Day on Saturday, June 24th. We're going to be from 1 to 4 p.m. in the Avon Senior Center. And it's really, it was a great event last year, and looking forward to it this year. And thanks to the archaeologists and the volunteers from the Institute of American Indian Studies and from FOSA, the Friends of the State Archaeology Office. So they're going to be participating with us. So we're going to have a good team there. So looking forward to seeing everybody next time. Thank you.
All right. Thank you very much, Heather. Have a good night,
Thanks,
everybody.
Heather.
Thanks so much.